Hi there! Today, I would like to introduce you to Diego Nobrega and his PhD supervisor, Herman Barkema. Diego is a Brazilian veterinarian working towards his PhD at the University of Calgary. He researches antimicrobial resistance on dairy farms, and today, we would like to share his research with you. On a dairy farm, antimicrobials, or antibiotics, are used to fight infections and keep animals healthy, similarly to how they are used in human medicine. However, after years of fighting off these bacteria, the emergence of antimicrobial resistance is becoming a concern, both for human and animal health and welfare. It has been suggested that the use of antimicrobials in livestock production should be decreased to prevent or delay this emergence of resistance. However, we must first determine what kind of change or decrease will lead to the most important and largest impact. Antimicrobials are used to treat bacterial infections and can be administered in two different ways. The first route of administration is local, directly to the area of infection. The second route of administration is called systemic, as the antibiotic travels through the whole body. The systemic route of administration is often given with a needle, either under the skin, into the muscle, or directly into the bloodstream. Diego thought the first step in looking at resistance on dairy farms would be to determine what types of antibiotics are used on farms, how many, and how they are administered. When an antibiotic is used on a dairy farm, the cow and the antibiotic are recorded in order to monitor who is sick and who is getting better. Diego used a second method whereby once a bottle of antimicrobials was empty, it was thrown into a designated trash can so he could monitor the amount used. Diego then compared the records with what he found in the trash cans. He found that as the number of written records increased, so too did the amount of antibiotics Diego found in the trash can. Further investigation found that farms with good record keeping also had cows with lower somatic cell counts. Diego was now able to see what type of antimicrobials and how many were being used on farm, but how did these relate to resistance? Diego took all the information he had collected from the dairy herds, which included the type of antimicrobials used, route administered, how much was used, and the type of resistance that was found on each farm, and looked at this all together. He found that the largest impact on antimicrobial resistance was the use of antimicrobials that were administered systemically. The more this route was used, the more resistance that was found. Additionally, these antimicrobials were the same class that humans use and that resistance has been found in. This was very exciting because now Diego had a message. Although it seems intuitive that reducing all antimicrobial use will reduce resistance, the major risk factor for resistance on farm is administering them systemically. Therefore, in the future, we need to look at how we can reduce our use of these systemic antimicrobials, but still keep our cows happy and healthy. The use of antimicrobials is an important aspect on farms, and used properly in tandem with other key management protocols can lead to happy and healthy cows. This, in turn, results in better quality milk and more of it. Antimicrobial resistance is a complex topic. However, with the collaboration of producers, veterinarians, doctors, and researchers, we can help keep our animals and humans we care about healthy. Thank you to Dairy Farmers of Canada, Alberta Innovates Technology Futures, and funders of the IRC in Infectious Diseases of Dairy Cattle. Our research team would also like to thank all the participating producers. Without you, this research would not have been possible.